Hello students, welcome to Capman Academy, and today we are going to discuss hurdle technology, right? And which is a very intelligent combination of combination of hurdles, or you can say the obstacles. These obstacles, they actually secures the food from pathogenic microbes and contaminants, which can actually lead to the diseases and harmful effect in humans, right? So in this intelligent combination, what happens? We give so much obstacles to the pathogenic microbes so that they finally cannot survive them, right? And due to this, our food gets secured. Its nutritional value remains as such. In this case, nutritional value of food, it does not change. Same way, economic viability of food product, it also does not change. Economical food product viability, food product viability also does not change. So which means that when we give the hurdle technology to the food products, it finally, the purpose of this technology is that it secures the food in which pathogenic microbes and contaminants all are removed, and the nutritional value, it remains as such, does not change, and also the economical viability of the food product also does not change, right? So the basic hurdles in the hurdle technology which is given to the food products are your temperature, number one, temperature. Second, water activity, water activity, which is denoted as AW, and it is the ratio between vapor pressure of the food itself, right? Another hurdle is the preservatives. Preservatives, in which we actually add chemicals so that our food remains for the longer time. For example, nitrite and sorbate. Another hurdle is called competitive bacteria addition. For example, lab bacteria, right? Same way, another hurdle is the acidity, which actually maintains the pH of the food products. So all these are the basic hurdles, as you can see in this image, that first we give the pH treatment to the food material, then we give the water activity, we maintain the water activity of the food material, and then we maintain the temperature. It can be high temperature or it can be low temperature treatment, right? So you can imagine that after going so much hurdles, there is no chance that pathogenic microbes will survive. So the ultimate purpose of the hurdle technology is to kill pathogenic microbes, right? And increase the shelf life of food material. Now, let's talk about some various novel hurdles. That is number 1, nano thermosonication. It is also used as a hurdle in the food industry. Second is the ultra high pressure, right? Third is the photodynamic inactivation. Fourth hurdle is your edible coating. Edible coating. Fifth hurdle is the modified atmosphere pressure. And sixth is treatment with the ethanol. Seventh hurdle is Maillard reaction in the food product. Maillard reaction in food products. So they are the novel hurdles which also used occasionally, depends on the food product, right? And the main objective of this technology is to number 1, ensure the safety of foods from what? From the pathogenic microbes. Pathogenic microbes, right? And second purpose is to prevent the food spoilage. Prevent food spoilage. Third objective is to increase the food quality. Fourth objective is to control the infection of the microbes and intoxication of microbes. And fifth objective is to increase the shelf life of food. And next objective is also to decrease the economic losses all over the world. Right? So these are the main objective of the hurdle technology. This is the reason it has been incorporated. So the various aspects of the hurdle technology, how it works actually, the important aspects of hurdle technology are hurdle technology are number 1, homeostasis, right? And homeostasis actually it stabilizes the living cells. It stabilizes the living cells by regulating temperature and acidity balance in the food material, right? And this one leads to the another aspect, which is the auto sterilization of foods, and this one leads to the decrease spore contamination in the food. And this one process is called, it is called metabolic exhaustion. Right? This is the second aspect of hurdle technology. Third aspect of hurdle technology is stress relaxation. Right? In this hurdle, multiple stresses are given to the food products. Multiple stresses are applied on foods, right? And in those foods, actually, the pathogens can be present, right? So upon the incorporation of these multiple stresses, this stress leads to the increase of HSP proteins, which are the heat shock proteins, and these heat shock proteins leads to the weaker pathogens, right? Weaker microbes in the food material. Another aspect of the hurdle technology is the multi-target food preservation. Multi-target food preservation, right? And this one, different targets, different type of targets like pH, temperature, enzyme system, water activity were used with microbes, or we can say competitive microbes, and this all multi-target preservation will leads to the weaker, it will leads to the weaker microbe homeostasis, right? So in the hurdle technology, it is very important to know its purpose, because its main purpose is to kill or to make the weaker microbes, so that when you actually consume those type of food products, you won't get affected or any type of disease, right? So on that basis, different type of hurdles which are used in the combinations are: number one, they could be the type of hurdles, they could be your physical hurdles, physical hurdles, right? And it could be treatment with the ionization, right? Treatment with temperature, it could be at high temperature or it could be at low temperature. Modified atmosphere, to be radiation treatment, or it could be electromagnetic radiation treatment. So they are your physical hurdles which are used in the food industry. For example, if we talk about the temperature hurdles, right? So in some processes, there is sterilization is used to kill the microbes, right? And with the sterilization combination, another process is used which is called blanching, right? And with the combination of blanching and sterilization, another process is used which is called pasteurization. Sterilization is used more than 100-degree temperature, whereas pasteurization is used between 60 to 85-degree centigrade. So all these are the temperature treatments given to the food material, so that food does not have any pathogenic microbes in it when finally it comes to your mouth, right? Same way, another type of the hurdle is physiochemical hurdle. Physiochemical hurdles, right? So they are nitrate, sodium nitrate, sodium sulfate, right? Sodium nitrite, sodium nitrite, then potassium sulfite, potassium sulfite, phosphates, salt, spices, gases, mixture, organic acid treatment. So they all contribute in the hurdle technology and even can be used as a physiochemical hurdle. Right? Another type of hurdle is your microbial hurdle. Microbial hurdle, in which there could be bacteriocins, 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 competitive microbes, competitive microbes which does not allow the growth of pathogenic microbes, and treatment with the antibiotics. Right? So these are the major type of the hurdles which are used in the food industry to kill the pathogenic microbes or to make them weak so they cannot grow on the food materials. Right? Now let's talk about the application of the hurdle technology. So the hurdle technology, it is very much used in the dairy industry. Dairy industry. Second, it is also used in the fruit and vegetable processing. Third, it is also used in the products which are derived from fruits, and also used in the meat industry. So this technology is very famous, and it is used in different type of food industries, right?